I see Luigi. Hello. Good morning. So
hello, good morning. I hope you can hear me.
We can.
And I hope that everybody feel, empowered by this incredible presentation. That really create the justification for everything that we are trying to do. I was going to introduce the people that will work in this panel. My name is Luigi Ferrucci. I am the scientific director of the National Institute of Aging at the National Institute of Health in Baltimore. And I've been involved in studying aging for a very, very long time. In fact, somebody suggested me when I was 15 that if I wanted to make something different in the world, I needed to study aging. And I followed that suggestion, all my career. And it's the people that know my work know that I'm incredibly enthusiastic about the new trend of finding biomarkers. But for once, I want to, direct this panel that will talk about phenotypes, because it's very important that we understand that why the biology will give us the key to realize the neuroscience dream. If we don't target, the phenotype, the disease, the condition that are associated with aging, all this research in biology will not be very usable. In fact, none of my very old patients ever say, I wish my mitochondria would be better. Or somebody say, I wish that my mTOR will be more inhibited. They say, I wish I could be at the wedding of my daughter and dance with my relatives. And that is the goal that we all have in mind. And I think that we should always remember that. So today we have 4 formidable people that will talk, and they work in our group. And Perry Kuo, who is a fellow, worked with me, will talk about how to create a multidimensional phenotypic marker of aging. I want to tell you why this is so important. If we want to slow down aging, if we don't know how to measure aging, we will never be able to demonstrate that we are slowing down aging. And then Lenore Launer will talk more about the cognitive part that can mention it. And then Aaron Simonsick will show you some of the pilot new phenotype that we are developing that capture some of the dimension of aging that have not been done before. And last but not least, certainly Toshiko Tanaka, he was a staff scientist in my group, will talk about how to connect a new technology that is proteomic to the phenotypic manifestation of aging. And I hope that you will enjoy, and there will be a lot of questions, and we will be more than happy to answer. And having said that, I don't want to waste more of your time, and I invite you to listen to Perry, who will give the first presentation.
Hi, everybody. I'm Perry. Thank you, Luigi, for your introduction. Can you guys see my screen?
Yes.
Okay. Yes. So I'm Perry. So I'm going to talk about how we can track the changes in physical and cognitive functions using a longitudinal phenotypic summary. And I have nothing to disclaim. So I want to show this slide that Dr. Ferrucci has published several years ago. This is mainly talking about we can think about aging as 3 different metrics of aging, which is biological aging, phenotypic aging, and functional aging. The key here is that biological aging may happen in the very beginning before we see any changes in the phenotypes. And the same thing that we may have some phenotypic declines much earlier than we observe a significant functional declines. And when we start thinking about to treat the patient when we have a severe declines, it may be already missed the golden period for us to prevent of the aging process or to slow down the aging process. So the question is that can we assess the rate of aging that before we experience accelerated functional decline? So can we really assess that how well our body is before we cannot move fast anymore and we cannot think clearly anymore? So that's the question that we are asking. Can we assess the rate of decline in this period of time. And why is this important? Because only when we can assess the rate of changes in the time before we have significant functional decline, they will help us to find the underlying biological mechanisms that predict what happens before we have this significant functional decline, so that we can potentially understand the potentially molecular mechanisms behind it. So we will have a framework slide later part to emphasize this point. Indeed, to do this, we need longitudinal data because, as the keynote speaker mentioned, if you ask everybody about the aging, everybody have different ideas. And it is that you check something continuous longitudinally, you really know that if it changes faster or slower. And Dr. Belsky and Dr. Elliott in 2019 cohort, they have demonstrated they select 19 phenotypes and they trace them across a period of time. Basically, in the first study, they traced them from 26 to 38. In the second study, they traced them from 26 to 45. And what they found is that if they average the rate of changes in these phenotypes across this 10 to 20 years of time, that it is associated with their rate of change, it is associated with their functional decline. And indeed, this is a still relatively younger group, and we know that sometimes rate of changes happened faster or slower in the later part of life. So to really address this question, in Baltimore longitudinal study, which is a study that we enroll people across a wide range of ages, and we measure their functions, phenotypes, and we also collect their blood for lots of omics research. But before we dive into the data, we have this conceptual framework, which means that we believe that there's a biological rule that drives all this aging, and they may be at a molecular level. And as time passes by, you may see some changes in your phenotypes. And we categorize phenotypes in 4 domains. The first one is body composition. It's basically about your muscle, your fat, your bone. And the second one is domain is about your energy. It's basically about how much energy you have and how you use the energy that you have. And third domain is homeostatic domains. It's mostly about autonomic function, insulin resistance, inflammation. And the fourth domain is about neurodegeneration domain or neuroplasticity domain. It's about your balance, about your brain, about your nerve conduction, things like that. And all of these can interact with stress, the environment, the social exposure, and lifestyle. And as time passes by, when you have some declines in these phenotypes, you may be able to compensate it, but at a certain stage, you just cannot compensate it, and you can see a functional decline. For example, you may feel like you don't have enough energy, you can still work fast, but at a certain point, you just cannot work fast as you can. So that's how we define the different levels of phenotypes and functional levels. And in our study, we spend a lot of time to collect the phenotypes representing these 4 domains in Baltimore longitudinal study, so that we can understand that if a person ages faster or slower in each phenotype. And we believe that to really capture aging, we should not cherry pick a specific phenotype. Instead, we should have this holistic approach, use this holistic approach to look at all phenotypes across 4 domains. So I'm going to give you an example of what I mean about the longitudinal approach. So I select one phenotype. This phenotype is called energy regulation or energy utilization. It's basically that we ask you to do a standard task, and we measure how much oxygen consumption that you need to achieve it. And then we also ask you to do more intense exercises, intense that you can achieve, so that we have a good understanding that how much energy that you may potentially have. And then we take a ratio between them. So that means like to finish a standard task, how much of energy that you need to accomplish that. And as you can imagine intuitively, as you get older, that the ratio is higher. That means that you cannot use energy as efficient as you are young. So here on the left-hand side, each single fainted line, it means that a person's the person's cost-capacity ratio at different time points. And then you can see that some people changes faster, some people changes slower. Because we collect data across a wide group of people, we can kind of estimate that people at your similar ages, what's the average rate of changes among the population that in your age. Then you can have more objective idea to say, okay, for this phenotype, this person like age faster and slower. So this is the approach we use, and it requires you have a wide range of people for you to build that reference. And on the right-hand side, we just want to highlight that the rate of changes may different across your age. And there's some people, most of people are like still very close to the mean, but there are always some outstanding people who change faster and some people who change slower. And this is just a slide showing the longitudinal changes of a phenotypes we collect across these 4 domains. And what we do is that we have this accelerated, we calculate the rate of changes of each person, and we reference it to people with the same sex and same age, and then we summarize the phenotypes within the specific domains. For example, in a body composition, we have their weight, we have their fat mass, we have their lean mass, and we measure the changes of length, and then we take the average of them. So we get this body composition specific longitudinal score. And then, as we mentioned, we hypothesize this is going to be 4 important domains representing these phenotypes. So this includes body composition, energetics, homeostatic mechanisms, and neuroplasticity or neurodegeneration domain. And then we average 4 of them to construct this global longitudinal phenotypic score. So next important question is to ask, like, how does this global longitudinal phenotypic score really capture the rate of changes in your functions? And we can see here that we look at the relationship between this score and changes in your physical functions. And we do see that a faster decline in their phenotypic score, longitudinal phenotypes, is associated with a faster decline in a gait speed, and longer time to finish 400 meters walk, and faster decline in your short performance physical batteries. So let me slow down a bit. I'm just going to focus on this first figure on the left-hand side. You can see the left-hand y-axis is the rate of changes in your gait speed, right? And then, as we know, get older, that you walk slower. And on the x-axis is basically that global longitudinal score. So you can see that those who decline faster are basically decline faster in the phenotypic scale are associated with those who decline tend to decline faster in their gait speed longitudinal. And then we also do the same thing. We look at what's the relationship between this global longitudinal phenotypic score and rate of declines in the physical functions. And we can see that this global longitudinal phenotypic score is associated with faster decline cognitive functions. And we also look at whether this score is associated with the pace of accumulation of multi-morbidity index. And we do see that if you have worse phenotypic profiles longitudinally, then that these people tend to have faster accumulation of their multiple morbidity index. And on the right panel, we also see that this score also predicts the mortality. So the key thing of this idea is that when we want to tackle aging, it's very important to measure aging before that we have some significant functional decline. And our conceptual framework conceptualizes phenotypes across 4 domains, and we measure the rate of changes of each phenotype longitudinally, give us a potential way to summarize a global longitudinal score to predict the rate of changes in the function level. So for next step, if we can further link this with the underlying biological omics data, then we can potentially figure out that what's the mechanism behind aging, and we can potentially target that. So just analog to the keynote speaker, just like if we focus on the what happened behind this polio, then we can potentially find a sort of device for analog to this Salk vaccine. But if we focus on the iron lung treatment, then we will never get there. And this whole research is about like how we create things for us to use as a tool to understand the underlying biological mechanisms. And I would like to appreciate all the people here. These data are in using the data from BOSA, so we also very appreciate participants to join the study for a long time. The QR code here, if you scan on it, you can link to the BOSA website. If you scan on the QR code on my slide, you can link to the papers we publish. And thank you.
Thank you very much, Perry. I think that that's a great introduction. I feel that it will be better to have the full presentation and then to have questions later on, just to make sure that we have time for everybody. So, Lenore, thank you for participating.
Yeah, so I'm going to talk about brain aging, looking at longitudinal and cross-sectional perspectives. So just to go, this is a very familiar picture suggesting that dementia begins at age 65, and there's logarithmic increases in the incidence of dementia as people age. So I'm going to talk a little bit about brain aging, aging of risk factors for brain aging, and metrics of accelerated brain aging. So many of us are familiar with the definition of dementia. It's basically a loss of cognitive function, as well as an impairment in occupational social function. But importantly, it requires a change from baseline. So there needs to be at least 1 or 2 longitudinal follow-ups to ascertain whether somebody really has dementia. But really, dementia is an age-related disease. It does not begin in older age, which I hope to show you. And for this, we need a life course perspective. So briefly, a very 2-second lesson in neurodevelopment. Obviously, the brain starts forming in the embryonic period, and by 2 years of age, one has the majority of neurons, although the brain obviously still keeps developing until your mid-20s. Over time, cognitive function begins to decline. In this cross-sectional analysis, it suggests that there's a couple that start to decline in your early 30s, and by your 60s, the majority of functions have started to decline. However, if you look longitudinally, then you can see that, in fact, the decline maybe begins a bit earlier in more functions, and that by age 50, there's a convergence in the fact that most different functions start to decline. There's also significant declines in the size of brain structures, and these structures significantly atrophy at different ages and rates. So total brain volume, there's a significant decline in your mid-30s to 60. The cerebral cortex a little bit later for the significant decline. White matter starts a little bit later and has a significant decline going into your 80s. And the hippocampus, which is very important for memory and also a key structure in Alzheimer's disease, begins declining at age 45 or 50, and really the decline is significant thereafter. There's also differences in the rates of decline amongst different sexes and different ages. These are data from the Cardia brain MRI sub-study. And you can see, for instance, that there's a difference between men and women in terms of total brain volume at age 55 years of age. But there's also significant race differences in when the pathology in the brain begins to appear. So Black men and Black women, there's significant increases in white matter lesion load compared to white men and white women. So looking cross-sectionally, there's of course a decline in cognitive function, as I've shown, or in brain volume. And when you look cross-sectionally, there's of course a mixture of people who are going to decline and those who aren't. When you look longitudinally, again, you get the advantage to see that there is an average decline, but there is also a subgroup that specifically declines in total brain volume at a quicker rate. And those are the people that are probably more at risk for Alzheimer's disease. So we know that there's a genetic contribution to dementia. There's been a lot of progress in the last few decades to try to understand what the different pathways are causing the dementia, looking through genetics. And for instance, we've learned a lot now to know that the immune responses are very important in the disease, but also there's some epigenetic changes that occur that may modifications in gene expression that may also contribute to dementia. So not only is the brain changing over time and different substructures in the brain are changing differently over time, but also environmental exposures, which also contribute to brain aging, show different associations depending on the age of measurement. So this is just a quick look. These are differences in risk factors amongst individuals going from their 20s to their 50s and 60s. You can see in the cardio group, there's an increase in the obesity over time. There's a big increase in diabetes over time. But these plots from the Framingham study also show you that these are not linear increases. That, for instance, specifically diastolic blood pressure is an example where you can see that up to age 50, there's an increase. And then after that, there's a decline, possibly reflecting arterial stiffness and changes in the hemodynamic system. These changes are not these changes are differential depending on whether or not you become demented. In this study, a 32-year prospective study in the Honolulu Asia Aging Study, you can see that individuals who are going to wind up being demented at the end of the study lose weight at a significantly faster rate than individuals who do not go on to dementia. And the differences are just as dramatic as in blood pressure, where there's individuals with Alzheimer's disease have a significantly faster decline in systolic blood pressure. And it's even more remarkable in individuals that go on to have vascular dementia. So the life course of brain aging in relationship to risk factors, based on the age, gene, environment Reykjavik study, we had birth records from midwives, and we found here that it's actually a pretty clear linear relationship between birth size, which is ponderal index, which is a measure of third trimester fat deposition, low PI indicating insufficient fat deposition in the third trimester. So there's a significant trend in relationship to total brain volume measured at on average 76 years of age. So birth size continues to carry through when looking at late life processing speed. We see that amongst individuals that had a low ponderal index, low education, people with a low education perform significantly poorer on the cognitive test, but amongst individuals who had a high BMI, the effective education was beneficial, and you can see that the difference in the scores between the high and low educated individuals is much lower, suggesting that environment can also subsequent exposures can also modify your risk and your performance. This, just carrying this theme through a little bit more, even vascular factors measured at midlife interact with birth size, and looking specifically here on total brain volume, you can see that individuals with low PI indexes and have a high risk level of cardiovascular risk factors also do more poorly, have lower brain volumes compared to the individuals that have none or one or the other. So not only is birth size important to take into account, but also the transition from midlife to late life. This slide shows a cross-sectional relationship between late life blood pressure and performance on a cognitive test. And you can see unexpectedly that there's it looks like high blood pressure is associated with low cognitive function. But if you look at blood pressure measured in midlife, there is an increase in risk for performing poorly amongst people that had high blood pressure, and particularly amongst individuals never treated for high blood pressure. And this trend carries through in terms of exposure to midlife, elevated levels of midlife blood pressure, and dementia, presence of amyloid plaques, and also the effect of treatment for blood pressure. So we like to think that there's a little change in the genetic code over time, but in fact, there are differences in gene expression at different times in tissues. So this slide shows, in fact, there's looking at 3 different genes. There's 3 different patterns of gene expression that vary from brain region to brain region, and also from cell type to cell type. So we are working on some time-integrated biomarkers of brain pathology, and in particular, I'll just show you the results of the epigenetic age clock that I'm sure will be discussed more, as well as MRI metrics of brain aging. So this slide shows you that that aging changes the methylation in neurons, and that this methylation decreases with age in those with no AD pathology in the neurons, versus not significantly. There's no significant change in neurons that are measured in individuals with a mild to severe Alzheimer's disease pathology. And we can see here that in mild to severe Alzheimer's disease, there's a bigger difference between the epigenetic and chronologic age, and that's significant. And this last slide, that the epigenetic age is higher than chronological age in the neurons of severe Alzheimer's cases, suggesting that most of the change in methylation will occur prior to the disease state, but there are also differences in individuals that are severely demented at this point. Here we have a look at a brain age metric by using MRI, looking at brain aging, and we can see that brain aging metrics, so the higher the brain age, the more significantly impaired, frail you are or aged, that there is an association between brain aging and whether or not you have ideal or elevated blood pressure. So putting these 2 metrics together, we can see that the epigenetic clock really does quite well in distinguishing individuals in terms of whether or not they did well on their global cognitive test at age 50, 55. Actually the metric based on the brain MRI does not do as well, as it does basically the same as looking at chronologic age. But when you combine both the EpiClock and the MRI metric, you get slightly better performance in being able to separate out individuals with poor cognitive status versus better cognitive status. And just to finish up, of course, the epigenetic changes aren't just different in the brain, but they're also different in relationship to different risk factors. And these data, again, from the Cardia study of middle-aged individuals, you can see that the epigenetic changes are most significantly noticed in individuals that smoke. And this, of course, is notable since smoking is a very big risk factor for cognitive impairment and dementia. So in summary, age is the major driver of brain aging. Brain structures and function decline at different rates over the lifespan. Environmental and genetic risk factors contribute to brain aging. Both genes and the environment change over time. Epigenetic age changes associate differently with different risk factors. epigenetic aging correlates with cognitive function and modestly correlates better with added MRI metric of brain aging. But these gene and environment changes and risk factors and in the brain need to be considered when investigating longitudinal phenotypes of aging and brain aging. And I will end there. Thank you very much for your attention.
Lenore Simonsick is the next speaker.
Yeah, so here I go with my attempted screen sharing. I'm going to be talking about metrics of aging of a different sort than Perry was sharing with you, a little bit more focused on the fitness end of things. So these are basically two works in progress, one expanding the way we look at fitness and the other expanding the way we look at fatigability, sort of two ends of the spectrum. So fitness, unfortunately, fitness decline with age is inevitable. This is data from the Baltimore longitudinal study of aging that uses VO2 max testing. And we can see that there is a precipitous decline and accelerated decline from age mid-30s to age 80. Oh, that's not right. Okay. And then this longitudinal change that accelerates with increasing age is independent of the loss of fat-free mass that is observed with aging. And it's also, unfortunately, independent of physical activity participation. So we're seeing the same rate of decline. It's just that those who are active or more highly active have a shift to the right. So they're starting at a higher place, but the rate of decline is the same. Again, all of this data comes from VO2 max testing, which is expensive and often impractical in many studies. We are fortunate in the BLSA to be able to do this. So for an alternative that is more potentially usable in population observational studies, we developed at this point over 25 years ago the long distance corridor walk. It was developed for the health aging and body composition study. And it consists of, in some cases, a 2-minute fast-paced walk or a 2.5-minute usual pace walk, which is the version in the BLSA, immediately followed by a 400-meter walk done as quickly as possible. And that's what we tell the participants. It's laid out, you don't need a treadmill. It's laid out on a course that's 20 meters long. A lap is 40 meters, and so 400 meters is 10 laps. So pretty simple. This is a measure where we have tested against max treadmill testing actually within the BLSA, and the correlation is excellent. It's at 0.8. And we also see with this test using data from Health ABC, and just a reminder that faster is better, so that decline actually will be going up because the time to do the test is going up. This also tracks with, there's decline, sensitive to decline over time with age, but also with regard to physical activity, so that people who exercise still decline in the 400-meter walk in the same way that they did in max treadmill testing. Okay, so here we are trying to develop an age normalized aging measure as an aging metric. So the source population are BLSA participants age 50 to 92. We're stopping at 92, even though we have participants all the way up to age 103, because the number of observations at 92 gets quite above age 92 is quite small. So this information is based on 600 men and about 650 women, and the observations range from 1 to 13 per person for 2,200 observations on men and 2,400 observations on women. So I'm going to draw your attention to the bottom of the graph, and you will see the age ranges are in bins of about 5 years around 50, 4 years around 60, 3 years around 70, and so on. And the bins are in incrementally smaller ranges of age to linearize to take out the curve in the accelerated decline. So the bottom two lines represents the 25th percentile of performance in the 400-meter walk for women, and the gray line is for men. And the women and men track together fairly closely, with the men doing better than the women. The next two lines are the 75th percentile for men and the 75th percentile for women. For women, the departure is more significant with increasingly older age. And it was good to hear from Ken as a reminder of the baby boom generation and what and how the world changed for women in that generation. So we can see or speculate that the accelerated decline in women in the oldest age groups is largely a factor of not having had opportunities to participate in stimulating exercise when they were growing up. Okay. So I want to focus two things. I looked at the group age 50 and older and age 70 and older. And I put a box around the mean age because this is age normalized. The mean age of those who we define as poor, those in the worst 25th percentile, average those in the middle 50th percentile, and those in the robust group are virtually the same. And the percent of men is the same because it's also sex specific. And just for proof of concept, we see that self-rated health is higher in the robust and lower in the poor. Obesity is rare in the robust, down to 7 and 5% respectively in those 50 and older and 70 and older, and well over half of those in the poor category, age 50 and plus. Sedentary behavior is highly common in those poor. It's also actually present in those with robust health at a level of about 30%. And the percent active is also about 30% in those robust and those poor health, or in age 50 and older and 70 and older, my apologies. So here we're looking at blood biomarkers for these three different groups. And this is sort of a proof of concept. And we're looking at adjusted means, and we're comparing these three different measures. The focus I want to draw the area I want to draw your attention to is the, let's say I can do this here, is the R versus age. So that's the robust versus average. Many of the metrics that we have that look at, I need to go back one. Many of the metrics that we have that look at aging really focus on or differentiate those in poor health and those in average to better health, with not differentiating the average to better very well. And so what I want to draw your attention to is this first column in the orange group, which is robust versus average. And so what we see for most of the blood markers, that the big differences are really driven by those in the poorest health. This is certainly true for fasting glucose and for free thyroxine and for the most part for inflammatory markers, with the exception of CRP, which does seem to flip the other way, which is much more differentiating of those from robust to average. Triglycerides works across the board, as well as white blood cell count. The next slide is for those 70 and older, or 70 to 92 more specifically, and the same trends are observed, where fasting glucose is really identifying people who are sick and is not different in those who are robust versus average. In here, we're looking at what I would call the non-blood biomarkers. So it's sort of more looking at each of the different systems. So we're looking at resting heart rate, where we see the difference is really driven. It's consistent across all three groups, but it's largely driven by those in the poorest health. This is very different when we look at lung function, FEV-Z score, where we see, and this is also an age-graded score, so there's a huge difference in each of these three groups. Body fatness is also remarkably different and expectedly different in the three groups based on weight-height ratio, as well as percent body fat derived from DEXA. The DSST is a digit symbol substitution test, And so we're seeing it's a measure of cognitive performance, and it's largely a time test. And we're seeing the robust group being much better than the average and the poor group across the board. The CSD, most people will know, is a measure of depressive symptomatology, where the cut point of 16 defines high levels of depressive symptomatology. Obviously, those levels are very low in this group, but it is very differentially expressed based on fitness level as a measure of an aging metric. Estimated GFR follows along the same lines. Pulse wave velocity, a measure of arterial stiffness, follows along the same lines, and that is also true of grip strength. And we see exactly the same relationships, with the exception of resting heart rate and pulse wave velocity, when we look at the older group, age 70 to 92. So we have a lot of more work to do here. So the next step is to bring in data from additional cohorts. Health, aging, and body composition in Chianti are cohorts supported by the Intramural research program, from which the BLSA is also from, and this will enable us to have more robust estimates. An important factor is that even though the 400-meter walk was designed to be more inclusive, to have fewer exclusions, we did have a lot of individuals who had a test failure. Either they were excluded because we had concerns about their initial health status and cardiovascular risk, or they started the test, but they stopped at different parts of the test. In order to include them in the study, we assigned them a time of 900 seconds, but I think we can refine that a bit to be able to include more of our 90-year-olds in our evaluation. And we want to revise and provide more age-normalized percentile gradations, and that includes a much larger sample, so we can have the 10th percentile, 20th percentile, and so forth. Okay, shifting to the other end of the spectrum, where we have a measure of fatigability, and this is susceptibility to fatigue. It's observable as performance deterioration or perceived exertion while performing a standardized activity. This has been underlined because that's what we've been doing in the BLSA for now I think it's almost 15 years. So the standardized activity that we use is walking 1.5 miles an hour, which is 0.67 meters per second for 5 minutes. And the exertion assessment is the Borg rating perceived exertion, which is shown in the chart to the right of that. It is found to have high levels of concurrent validity and high levels of predictive validity. And we have published a bazillion papers showing the relationship between fatigability and various outcomes, and how it is more sensitive than straight measures of fatigue, as individuals modify their behavior to stay below their fatigue level. But by stressing them a little, we are more able to capture their fatigue. But this test may not be stressful enough. So our RPE score, when we look at this test, we can see it has a really low measurement ceiling. So this is the distribution of BLSA participants aged 65 to 75 years. And most people are well under 11. And frankly, most people are hanging out at 7 and 8. You see the odd distribution here that goes up and down. That's because we learned after the fact that people are more inclined to give us words instead of numbers. So we've developed a higher level test, we hope, where we are looking at stage 1 is exactly the same as the original test, but then we've added a stage 2 and stage 3. Stage 2 is where there's a gradual increase to an individual's own usual gait speed for 5 minutes, and that usually happens in the first 1 to 2 minutes of their 5, and then we ask their Borg rating after the next 5 minutes. And then in stage 3, they continue at their usual gait speed for additional 5 minutes, and then we have their exertional assessment. So basically, this is trying to understand, can somebody walk for 15 minutes at their own self-selected pace, and then how hard is that for them to do? And this hopefully will allow us to look at this metric in much older age groups. And so this test was implemented basically 4 months ago in the BLSA. So we don't have a lot of data to show, but I was able to ask you a few favors, get data that had just been entered. And so we're looking at, in the blue, the standardized activity, the original test, and the distribution actually is a little bit different than the work I had just shown you, because it's the maximum that we're seeing, or the most common level we're seeing is 11. This is in part because this is our post-COVID BLSA participants, and we were trying to bring in the oldest and most frail because we wanted to continue to follow them and their progress and their decline. We can see, though, when we go to the next stage, everything does shift in the way that we had hoped, so that the more common scores are now 11 and 13, and we're all the way up to the level where people have stopped. And then the additional 5 minutes again shifts the scores up to a common level of 13, and then even all the way up to 17. So we have a lot more to do here. We have a lot more data to collect. We need to also include data in more robust and younger BLSA participants to see if they also show increasing levels of reported fatigability with longer workload. We need to conduct added value studies. Are we really learning much more by using this metric in addition to all the other ones that we have? And we want to evaluate its potential as an aging metric among the oldest old, particularly those individuals who are really not able to do a full 400-meter walk test. And with that, thank you for listening.
Thank you, Eleanor.
Stop sharing.
We are a little bit late, so I will just ask Toshiko to start presenting. And I want to tell everybody that if they have questions, please put that in the chat. If we have time, we will give you an answer, but I'm sure that the speaker will be more than happy to provide you an answer by email or during the meeting through the chat.
Thank you. I'm going to try to move through this so we have some time for questions. I'm going to be presenting some of our studies looking at the plasma proteomic profile of aging. So one of the motivation to do these kind of studies is because we know that advanced age is one of the major risk factor for many chronic diseases. However, when we traditionally study diseases, especially in epidemiological study, we often control or ignore the effect of age. And the goal of geroscience is really to aim at understanding the basic biology of aging, to uncover the common mechanisms underlying aging and age-related diseases. And one way to get at mechanism is to use different omics data. And our previous presenters have talked about different omics data, including epigenetics. But in these studies, we're focusing on proteomics. And proteomics is particularly attractive because it is a common thing that's used in the clinic. We already use proteins to assess disease risk. So there is great potential for translation, and also interpreting results from proteomic studies is straightforward because proteins are close to the molecular function. One of the challenges of studying proteomics and aging is that many chronic diseases, the prevalence increase with age. And so it's really hard to separate the effect of disease and aging when we look at a general aged population. A second challenge is that blood is an ideal tissue to study proteomics, but due to the dynamic range of proteins, it's difficult to measure a large number of proteins. So we've tried to address some of these issues by utilizing cohorts that are free of diseases. And also we've used recent new technology that has targeted proteomics that measure thousands of proteins in a small amount of blood. So, okay, so these are the two studies that I'll be presenting. These are two populations. The first one we looked at disease-free healthy participants. This is the study to the left. And then we followed this up, the first study, with a second study looking at a more general population of older adults. The first study, we used a cohort, the Baltimore Longitudinal Study of Aging, or BLSA, and the Gestalt study, the genetic and epigenetic signatures of translational aging and laboratory testing study. These two studies are based in Baltimore using a very similar protocol. We used data from 240 healthy subjects ranging in age from 22 to 93 years of age. And again, these are very healthy population that had no chronic diseases with the exception of controlled hypertension and no cognitive impairment. The second study was done in the Chianti study, which is based in Italy. We used the baseline sample that was performed between 1998 and 2000. We had 997 subjects ranging from 21 to 98 years of age. Unlike the BLSA and Gestalt population here, this was a general population of older adults, so there were definitely people with some chronic diseases. For the proteomic assessment, the two studies was performed using the same assay. This is the SomaScan assay. We used the 1.3K version, so we measured about 1,300 proteins. One advantage of proteomic assessment is that we're able to leverage stored samples. So most of our samples came within 5 years of proteomic assessment, but we did dig into some of our older samples to be able to identify individuals that did not have any chronic conditions. So we performed some quality control. We removed outliers from our proteomic assessment, which is expressed in relative fluorescent units. And for a univariate analysis, we used a linear regression. And we also created a proteomic clock using elastic net regression. So these are the results from our analysis, univariate analysis. On the left, we have the super healthy population of BLSA and Gestalt. And on our right, we have the Chianti population, which is a more general population. Each of these dots represent a result from a regression analysis. So there should be 1,300 dots here. The dots on the right here are proteins that are found in higher abundance in older age, and the proteins on the left here are proteins that are found in lower abundance with older age. In the BLSA and Gestalt study, we found 217 proteins that were associated with age, and in the Chianti study, we found 412 proteins that were associated with age. And one of the remarkable things that we found is that in the two geographically different populations, there was a high degree of consistency. For example, among the 217 proteins that were associated with age in the BLSA, we found 97% was replicated in the Chianti. Similarly, of the proteins that were associated in the Chianti study, about 80% was replicated in the BLSA. When we use the elastic net regression to create a proteomic clock, the first thing that we found is that we are able to estimate chronological age with high degree of accuracy using proteomics data. In this analysis, we have a 76 protein proteomic clock, and this here on the y-axis is the protein age that's predicted using the proteomic data and the actual observed chronological age. And what we see is high degree of agreement here. This clock was generated in the BLSA and Gestalt, the healthy population. And when we applied this algorithm to the Chianti, the general population, we do again see a correlation. But one thing that was very interesting that we observed is that within the Chianti, when we look at the healthy population that looks a lot like the BLSA and Gestalt, the correlation is much higher. But when we look at people with comorbidities who also tended to be older, the predicted age tended to be higher than the actual age. This led us to ask whether this proteomic clock contains information about biology. Checking the time here. And here we created metrics where we looked at the deviation between the predicted and the actual age, and those who were predicted to be older using the proteomics data had higher risk of mortality and accumulated morbidity quicker. So I'll just go to the conclusions here. So with the advancement of proteomic technology, we're able to identify proteomic biomarkers that are replicated across various studies. And also we could create a clock, much like the DNA methylation clock, that harbors information about biological age. So that was a little bit quick, but I'd like to acknowledge my coworkers and also participants of all the studies that we use the data for. So I think that gives us a little bit, maybe a minute or two for questions.
Yes, we have really short time, 1 minute or two for questions. There was a question in the chat for Lenore Launer about the rate of decline of cognitive function in male and female, men and women. Can you comment on that, whether there is any evidence that they behave differently?
I can't really comment on studies that have looked at differences by sex over time, but we do know that there are differences in sex in terms of cognitive function, different cognitive functions. So and also looking at the differences in the decline in total brain volume and looking at other MRI characteristics, as well as differences in the rates of chronic disease and dementia, I would suspect that there would be differences by sex in different cognitive functions looking prospectively. So there may be such studies, but I'm not aware of them.
Great.
Luigi, can I ask a question? And I think you might have noticed you have another 5 minutes. We can deal with that. But I'm a layperson, and I'm listening to this fascinating data about the phenotypic and markers. What is the relationship between the often spoken about molecular markers, biomarkers of aging, and what your panel has presented? Longitudinal phenotypic markers. Is one better than the other? It's a combination of things. How would you? Yes.
I think that's a great question, and I think that what do we know is that when you look at the phenotypic manifestation of aging longitudinally, so the same person that is measured multiple times, and then you look at trajectory of change, the association with biomarker is much stronger and much more robust. And one thing that I wanted to outline is that why it is important that we find this. When we'll start testing this drug that theoretically slow down aging, in order for us to do a clinical trial, we will need to wait 40 years until the population that we have tested dies. And of course, this is totally impractical, and no regulatory agency will ever accept that. But if we pin down few biomarkers of aging that are robust to predict longitudinal decline, and so they are good biomarkers of the rate of aging, and we can show that this intervention reduce the rate of aging, then we know that we have accomplished something. I think that Ken made a very incredible, to me, simple but incredibly powerful statement. With the aging of the population, we have lost health. In fact, increase in life expectancy have been much more, evident than the increase in health expectancy. So we want back that health that we have lost over time. And really the geroscience, it's important to accomplish this goal. Hit the disease when they're still not clinically manifest. We keep doing better for disease when we discover them, but the only thing that we're doing is prolonging the life of those who are sick. If we don't go before that, we don't prevent the onset of disease, we will not increase health expectancy.
Well, Luigi, is there anything more, or we?
Well, I wanted to. Toshiko didn't say something. I wanted to ask her one thing. So proteomics is really promising. I think that, as you mentioned, doctors have used it for many years. So, what is the relationship between proteomics and other omics, and what do you think is the future? Is combining omics, use them separately, putting them together? Do you think that we will increase our predictive value by, combining?
Yes, we hope so. So that was the two slides that I skipped over. So there's been a lot of clocks that are being made with different omics data. And I think one of the most familiar ones are the epigenetic clock. So when we compare the proteomic clock with the epigenetic clock, what we see is that when we compare the clock that was made using the similar method that we did, so basically tuned on chronological age, the performance of the proteomic clock is similar to the epigenetic clock. But then there are second and third generation epigenetic clocks that are tuned on phenotypes, and those seem to predict outcomes better than the proteomic clock. So that means that I think we have to be a little bit more creative. The proteomics and all these omics data do capture some information about biological age, but we need a little bit better algorithms. And I think these are being generated on a weekly basis. There's always a new data that comes out. And I think that we need to figure out how to layer these different omics data. And I think people are working on these algorithms where we combine epigenetics, proteomics, metabolomics. We're generating the data. We just need to figure out how to put it all together.
Thank you so much. I hope that this presentation have tantalized your mind and open a lot of synapses and make you think that yes, omics are important, but if we don't get a good phenotype or reference, they will not be extremely useful. So it's important that the two fields work together to create, that type of metric that we need to advance research on aging.
Luigi, I want to thank you and your panel very much for this excellent session.